everyone, and thanks for tuning in to BEaTS Research Radio, a podcast and YouTube channel dedicated to bringing our community closer to research in the science and engineering fields. I'm Hallie Arnott, a biomedical engineering master's student at the University of Ottawa, and I'll be your host on today's episode. With us is Dr. Marc-André Langlois, a professor in the Department of Biochemistry, Microbiology, and Immunology with the Faculty of Medicine at the University of Ottawa, and the director of the university's biocontainment facility. Dr. Langlois is also a Canadian research chair in molecular virology and intrinsic immunity. As most of us are now aware, due to the global impact of COVID-19, the spread of viruses between animals and humans can have very devastating consequences. Dr. Langlois and his research team are exploring ways to fight and prevent these types of infections. His research will lead to the development of diagnostic tools to identify new retroviruses that cause disease, as well as therapeutic approaches for treating infection. Dr. Langlois is also the principal investigator of the study Stop the Spread Ottawa in collaboration with the Ottawa Hospital Research Institute, and he's going to give us some insight on that today. Thank you so much, Dr. Langlois, for joining us here on BEaTS Research Radio.
Well, thank you for the invitation.
So before we delve into our pandemic topic, can you share with us what research goes on in your lab and what you've been working on with respect to virus biology for many years now?
So I'm a molecular virologist. These are scientists that are interested in how viruses replicate, how immune cells and host cells protect themselves against viruses. And I study this interplay between viruses and host cells and this never-ending arms race to infect or protect against viruses. So I've been working for a number of years now, over 12 years, on HIV and studying how host cells protect us against retroviruses and how retroviruses get around those defenses. In more recent times, I've been diversifying this research to influenza. And now since the pandemic, a lot of my research has been focused on the coronavirus.
Right, of course, like many labs now in virus biology. Since you're a virologist and someone who's worked with these viruses for many years, could you explain to us briefly, from your perspective, what's so special about and unique about the SARS-CoV-2 virus?
So what's unique about it is that we're dealing with a very infectious virus. It's a respiratory virus that is easy to transmit. It's a virus that can infect anyone. Everyone is susceptible to being infected by this virus. And for those that do survive the infection, most of us do survive, what we don't know are the long-term effects of being infected with this virus. We know that there's increasing problems that we are seeing in those that have been infected by the SARS-CoV-2 virus. Right.
That's a very good point to bring up. There are a lot of studies now coming out. It's like what I think there's one I was speaking to somebody with recently about what can happen to cardiac effects later on in life, which is something that is important. And something that you're also working on towards understanding this virus a little bit more is the Stop the Spread Ottawa study in collaboration with the Ottawa Hospital Research Institute. Can you tell us a little bit about the study and where it's at right now?
Certainly. So Stop the Spread Ottawa is a study to look at the long-term immunity to the viral infection, to the coronavirus infection. So when we're exposed to the virus, we develop antibodies, we develop some T cells that are activated that can recognize the virus. And what we're trying to figure out is how long this protection lasts. So antibodies protect us against new infections, reinfections, the same with our T cells. But over time, these antibodies diminish, start waning, and this is totally normal. What we want to know is how long will we be protected if we've been exposed to the virus? So that is the main aim of the study. The design of the study is to have 500 individuals that have been previously infected by the coronavirus. This is our convalescent cohort. And we'll be tracking these individuals every month for 10 months. We'll be tracking to see if there's reinfection. So we're going to monitor if there's viral RNA in their saliva. And we're also going to monitor their antibody response, and more specifically, their neutralizing antibodies, to see how fast these antibodies disappear. And with that, we'll be able to predict if those individuals are still protected or unlikely to be reinfected with the virus after 10 months. The study also is comprised of another cohort, a surveillance cohort. These are 500 individuals that are at risk of getting infected. These are individuals that are in contact with the public. We're talking about teachers, we can talk about taxi drivers, anyone, frontline medical workers, of course, that are at high risk of getting the virus. So we will be recruiting 500 of these individuals, and we'll be tracking them for, again, 10 months. Once a month, we'll be asking these individuals to provide saliva and a few drops of their blood in the form of dried blood spots. These will be collected at home. We'll be providing them with home collection kits so they can provide the saliva and the blood at home, and just mail in their samples or drop them off at the university. Then we'll be analyzing these to see if there's a presence of virus and presence of antibodies. As you know, there's a certain number of people that can be infected with the virus, but do not display disease symptoms. We call these asymptomatic carriers of the virus. And these individuals are responsible for spreading the infection, and they don't know they're infected and they're spreading the virus. So by looking for antibodies and the presence of viral RNA, we can help stop the spread and also study what makes these individuals so special. Why are they protected and not others? So these are the types of questions we'll attempt to answer with our study.
Yeah, that's very interesting. That's a lot of questions that I think a lot of people would like answers to as well. Do you have a hypothesis on what you think your study results, what you might find?
So what we'll find is we'll see a gradual reduction in the number of antibodies in those that were exposed. This is starting to be more and more documented now in the published literature. So we do expect these antibody levels to go down over time. But what we're really interested in is looking at the neutralizing antibodies. So these are the antibodies that will bind the virus and prevent the virus from entering the cells. And we'll be monitoring those specific antibodies over time. And this will give us a clue to how long the immunity lasts. And it will also help inform the vaccine schedules. So when the vaccines will be coming out, presumably in the spring, we'll probably need more than one shot. We'll need the primary shot and also some booster shots to keep that immunity against the virus. But what will be the schedule? Do we need a booster shot every 3 months, every 6 months, every 12 months? Well, this will depend on what we see with how the neutralizing antibody levels are maintained over time. We'll also be looking at T-cell responses because there are two arms to the immune responses. There's the humoral arm that where antibodies are produced, and there's the cellular arm where T-cells come into play and attack cells that are infected with the virus. So we'll be monitoring the T-cells as well in our study.
Right. So you also mentioned previously that what makes certain people carriers, but not, I guess, present any sort of symptoms. Is that what's that difference that you think is there between somebody who is a carrier and somebody who will get the coronavirus and be, yeah?
So there are no answers right now to that question, but we have suspicions. So some of the suspicions are that the coronavirus that's going around is not the only coronavirus. There are 7 human coronaviruses. 4 of them cause the seasonal cold. Now, when you're infected with a seasonal coronavirus, and we've all been infected with one of the seasonals during our lifetime, will those antibodies against seasonal coronaviruses help protect us a little bit against this SARS-CoV-2 virus? We don't have a clear answer to that. But it may very well be the case. Then come in genetic factors, and then comorbidities. Are the individuals being infected very healthy, or do they have underlying health problems that might aggravate disease symptoms?
That's very interesting. I'd definitely like to hear the answer for that one soon. So for this study, you mentioned you have the 500 participants, and then the other 500 participants are in the surveillance cohort. For that surveillance cohort, those are 500 participants who do not have the coronavirus. So with that, how does that change if one of those participants might contract the virus?
So what we would do is we would approach that participant, inform them they've tested positive for the viral RNA and are therefore possibly and very likely infectious. We'd ask them to self-isolate and get a clinically approved test to confirm the diagnosis. And then we would invite those individuals that test positive to be part of our convalescent cohort. And this way, we would continue doing our blood draws and monitoring the viral RNA. And this would provide some very important information concerning the very early stages of the infection. What happens when we're just infected? How does the immune system respond in the first hours and first days of the infection? We'll be able to track this with these individuals that were unfortunate enough to become infected.
Yes, that I guess is a negative that turns into a positive for the study, which would be an interesting piece of information. And from what I understand, you are still currently recruiting participants for the study?
Yes, so we started recruiting just over a week ago, and we have about 450 participants so far. We're looking for a total of 1,000 participants, 500 in the surveillance group, 500 in the convalescent group. Mm-hmm. And this recruitment is ongoing. So if any of you out there have been infected and are interested in being part of our study and helping us to better understand this virus, well, I welcome you to register to our study. And if you are someone who is working with the public who is at high risk of getting the virus, well, we'll provide surveillance for you once a month, looking at for the viral RNA and for antibodies as well.
Okay, that's good. Well, definitely for our listeners, anyone that's interested, we will put the link to the study so you can register if you would like, and we'll just link that below if you're interested. So about the results of this study in the, I guess, 10 months to a year, when they might be available, will they be open access, shared with other researchers who are currently working in the field, or also shared with the public who are interested in knowing the results of the study?
Absolutely. So in one perspective, we are sharing with the participants their status, so they can access a secured web portal where they can see their results for the antibodies. Of course, if they become positive for the viral RNA, they will be informed immediately to limit the spread. And for the general population and for the scientific community, we won't wait till the very end of the study to provide the results. We will be providing some data as we go along if we feel that information is helpful to better diagnose, to better treat the virus, and to better inform about the progression of the immune responses. So we will be giving regular updates.
So coming to that point with sharing your data and results of the study with other researchers who are working in the space, do you feel that sense of community and kind of global community, I guess, when it comes to studying this virus and working towards understanding it? Is there good communication globally or even nationally, I guess, with the research that's currently ongoing?
Yes, absolutely. There's a tremendous collegiality during the course of this pandemic between researchers across the globe to share information as soon as they have that information. We've never seen the use of preprint servers to this extent. As soon as studies are completed, researchers are putting the data up so that other labs can learn something about the virus immediately and not wait several months until something is published. We get to see the raw data, the first draft of the study, and this is extremely helpful. We also see a lot of labs sharing reagents, sharing protocols, and this has never been seen before, not to this scale. Of course, there's still some competition out there because we want to be the first to discover something and to show off the results. But overall, there's been tremendous collegiality between researchers in the field of the coronavirus.
Right. Yes, and I think it's something that is hopefully going to spread even outside of the realm of research of virology, because I think sharing and having that communicative collaboration too with other researchers in the field is always beneficial. For a final question, what do you think the key players are in order to be successful in fighting and preventing retroviral infections, such as the ones involved with COVID-19?
Well, just a small point, COVID-19 and coronavirus are not retroviruses.
Okay.
They're a different type of retrovirus. They're
okay.
Positive sense virus, RNA viruses.
I learned something new today.
What is key is to be mindful of public health advice. Public health is trying their best to limit the spread of the coronavirus, and their advice is critical to prevent an overflow of cases entering the community and propagating in the community, and also overwhelming our healthcare resources. So when public health says, well, we should all be wearing masks, there's science to back those claims. And we should be listening to those pieces of advice that are absolutely essential if we want to come out of this with a minimal harm to our economy and to our society.
Right, definitely. Well, that concludes our interview today. Thank you so much, Dr. Langlois, for joining us here on BEaTS Research Radio and speaking about the incredible research being conducted in your lab, as well as with the Stop the Spread Ottawa study.
It was my pleasure. Thank you for having me on your show.
Thank you so much to the University of Ottawa Heart Institute, the BEaTS Research Lab, and the Department of Biochemistry, Microbiology, and Immunology with the Faculty of Medicine at the University of Ottawa, who are all supporting us here at BEaTS Research Radio. Our director is Dr. Emilio Alarcon, and thank you to all of our listeners for tuning in, and we'll catch you all next time.